Okay, so we're going to talk about two things today. First, I'll spend the first half talking about motor cortex, because we haven't talked about, we've talked about sort of motor stuff further down, but we haven't talked about motor cortex yet. And then I will do a quick sort of midterm review of all the main points of the visual, somatosensory, and auditory systems. So motor cortex. So if we look at humans, so a lot of the work, of course, has been done in macaque monkeys, and so I always like to sort of put both, humans and macaques together so that we can sort of make sure that we're on the same page where stuff is. So here's the central sulcus in a human, and if we look in frontal sulcus, in frontal sulci, there tend to be sort of like two sulci like that. And then when we look at the posterior sulci, we see, the STS like that. So these are the ones to keep in mind. So we've got central and STS. And if we look at a macaque monkey, it's a smaller brain. Looks something like this. And the central sulcus, there's the central sulcus. And if we look in the frontal sulci, you've got the principal sulcus and the arcuate. And let me just label those. So this is called the principal, if you want to look at the, and that one is called the arcuate. And what happens in humans, you can sort of think of like, if you took the principal sulcus and the upper arm of the arcuate, that's kind of more or less equivalent to this guy, and then this guy is more or less equivalent to the bottom arm of the arcuate. And there's an STS, big STS here coming up in the macaque monkey, STS. So where is the motor cortex? So the motor cortex, it's basically in front of the central sulcus. So put central in here, central. So it's in front, and it kind of extends into the arcuate. And then it goes over onto the midline. That's supposed to be a cutoff arrow that's cut off by the midline. So it continues onto the midline like that. And pretty much the same thing in humans. So you've got sort of face motor cortex here, and the frontal eye fields somewhere up around there. It's just more or less in the same place. And it goes onto the midline, and there's, a substantial chunk of motor areas on the midline. They go a little bit anterior on the midline also, something like that. So that's what we're talking about. So let's now consider what's in motor cortex. And so there's been a debate, over many years about what does motor cortex do? Does it actually represent positions in space? So do different parts of the motor cortex represent kind of like the deep colliculus? Does it represent sort of different points in space, or does it just have a map of muscles? So maybe there's just a map, maybe there's just individual muscles, like there's a part for the biceps and a part for the triceps, or those muscles have several subparts. Are there sort of several are there just basically sort of representation of individual muscles? And why has this been a debate so long? Well, the problem is, in the motor system, a lot of times people are just recording where the animal's hand is, which is, of course, very important, because the motor system, the whole idea of the motor system is to put the hand in different places. integrate it with visual information, grab stuff, So that's an important feature to record, but also another important feature to record is which muscles are doing it. So if I grab something like that, well, that's kind of a complicated thing. It's involving my wrist, it's involving, my shoulder muscles, it's involving my triceps, the one on the back to make the arm extend out. So you can see it's a complicated thing because that's not straightforward to figure out which muscle is active. And the way that you do it, I mentioned it before, but you do something called EMG and electromyography. That's what it means, electro, sort of myo for muscle. And how do you do it? So if here's your biceps, it's actually painless, relatively painless. And so you just basically inject a little wire into the muscle. So the syringe isn't anywhere near this big, but I'll make it really big so I can draw the wire. And so basically you just have a fine little wire, and you put it in the syringe and bend the tip up into a little hook, like that. And then you inject that into the muscle, pull the syringe out, and then you got a little wire, tiny little fine wire, going into your muscle. And what does that record? Well, how does your muscle work? Well, a motor neuron comes down and it makes this end plate on your muscle, which is a giant synapse. It's like 200 times bigger than a little synaptic bouton up in the cortex. And what does that do? Well, that just causes your muscle to spike. So when an axon potential comes down here going that way, it basically just causes a spike to spread out across the muscle. So this spike will just sort of spread out across the muscle. And that's what causes the muscle to contract. But that's also what you can record with this electrode. So what you're seeing are basically motor neuron spikes with EMG. That's actually what you're actually just you're literally recording the spikes that the motor neuron sent to the muscle. The problem is there's a lot of different muscles, right? There's shoulder muscles, like 5 or 8 of them are on the shoulder, and then you got the triceps and the biceps. And so a lot of times people don't bother recording it, but I always draw a fish here. So there's some fish lips, and there's a fish, like an aquarium fish. There's a little nose. So people have actually figured out how suction feeding works in fish. So if you look at there's the arm of the fish, the pectoral fin. So if you look at a teleost fish, is a bony fish, like a salmon or something like that. They've developed this elaborate system. So if you look at a shark, so here's a shark. So, they got lots of teeth, but when they open their mouth, the mouth it's like an alligator, it just sort of opens the mouth like that. And it's not very good at causing suction. It's good for biting stuff, but it isn't good for suction. And so what teleosts did, so sharks evolved before teleosts did, so what bony fish, these are cartilaginous fish, so bony fish did is they developed the system of about 20 muscles that actually block off the side of the mouth. So if you've ever seen like a goldfish or another fish sort of sucking stuff up off the ground, but they can do that to another fish. Like a predatory fish will open its mouth, and then all these muscles will cause the little side of the mouth to get blocked off, and so you get a nice tube, and then you can suck the fish in. So enterprising researchers have taken teeny, tiny electrodes and put them in all 20 muscles of the teleost, and then, of course, the fish wasn't incredibly happy, but then they eventually got it to sort of eat something, and then they could record this complicated pattern of motor spikes coming out of all these different motor neurons that actually causes this behavior. Now, people haven't done that as much in the case of studying the motor system. They should do it more, because that's the actual final output answer. Like, what did all the muscles do when you did something? And because they haven't done that very much, there still is this debate, is there just muscles up here, or does this thing actually sort of code different locations in space? The answer is still not entirely clear. It's probably something like coding positions, more or less coding positions in space. But let's just now mention one thing about mice, just as a quick aside. So, because if you ever read any neurobiological literature, you'll often sort of, end up, reading about mice, because especially in the last 15 years in neuroscience, people have gone and done a lot of work on mice. They don't have a very good visual system. They got a good somatosensory system. But if you look at a mouse, what you see is the motor cortex is a little different than in a primate. So if we look at a primate or a cat, we know that the principal sensory nucleus of nerve 5, principal sensory nucleus, and the cuneate, this may be on the test, the cuneate, that's the arm, and the gracile, remember that one was the foot and the lower part of the body. Those nuclei project up, and they project up to, so they get input from the spinal cord, and then they project up to a thalamic nucleus called ventrobasal. That's got ventral posterior lateral and ventral posterior medial. You don't have to remember that. And so this is ventrobasal thalamus. And then that goes up to somatosensory cortex. And somatosensory cortex is area 3B and area 1. But it turns out there's another pathway relevant to the motor system that projects to a different nucleus called ventrolateral nucleus. And that one projects up to motor cortex, area 4. That's motor cortex. So that's and then these guys project back to each other. So the somatosensory cortex projects to motor cortex, and motor cortex projects to somatosensory cortex, but they're separate. So that's the situation in a monkey, cat. It's even the situation in like an opossum, which predates mice. But if we look at rodents are kind of they're very successful, but they've cut some corners, and so one place they have relatively small brains. What they've done with their somatosensory system is they've got a ventrolateral, and they got a ventrobasal, just like they do in primates. But when you go up to their somatosensory cortex, it's called somatomotor cortex. Both of these project to the same thing. And when you actually look at the somatomotor cortex in a rodent, it kind of looks a little bit like a combination of area 3B and area 4, because area 3B has a big layer 4, sorry about the 4 and 4, it has a big layer 4 because that's where the input comes in. You remember that there's more input to layer 4. And then the big motor output comes out of layer 5. So if you look at area 4, motor cortex, it's got a really big layer 5 because that's all the motor neurons going down to the spinal cord. If you look at somatomotor cortex in the rodents, it's got a big layer 5 because it's motor cortex, and it's got a big layer 4 because that's where all the input's coming in. So it's kind of strange, but I just thought I would mention that, that if in the unlikely event that you decide to breach the somatomotor literature and rodents so that you're oriented. So there's this difference about it. Okay, so let's put that aside now and just go to sort of the basic layout of this region here. And so basically what we have is we've got kind of a big patch like this of area 4, and it's more or less divided, into face, hand, and sorry, backwards. How can I do that? So foot is medial, and this is about the midline. So the foot slightly goes over the midline, sort of bends over the mid. I just flattened everything out. So here's face, hand, foot, hand, and face this way. And so what is that guy? That guy is, M1 or, area 4. Okay, so that's M1, primary motor cortex. So if we look in front of it, then we have premotor cortex. And so premotor cortex, you could maybe divide it into sort of two chunks. So this guy is premotor cortex. This is area 6. Why don't they put them in order? Yeah, but anyway, it's so where is these in relation to the other areas? So there's an area called 3A, and then that gets back into what we remember from 3B and area 1. So 3A is an interesting area because it has especially a lot of muscle. It's arranged into foot, hand, and face also, but it has muscle receptors in it. So a lot of, like muscle spindles, Golgi. So it has muscle. It has touch in it also, but it's got a lot of muscle receptors. Okay, so that's what we did when we talked a long time ago about somatosensory. Okay, so we got area 6, and some people distinguish this premotor ventral. And then if we look at, we talked about the frontal eye fields, remember the FEF, the frontal eye fields. And so the frontal eye fields are kind of sitting somewhere about here. They kind of stick out a little bit like that. And they're kind of divided into parts that are more interested in saccades and parts that are more interested in pursuit. Remember smooth pursuit. And those are, you can sort of think of that as basically sort of central versus peripheral, because saccades, you're mainly looking at stuff out in the periphery. Pursuit, you're sort of focusing on, let's just try to keep following something. So that's the frontal eye fields. So it's the F frontal eye fields. It's often just called the FEF. And then there's another, there's only a couple more. There's another area that's kind of extends out here. It probably has 2 parts in it, but this one is called dorsolateral prefrontal cortex. So I'll write that out. So what does that mean? So we have area 6, which is premotor. That's different than prefrontal. So prefrontal is something that's in front of premotor. So this is dorsolateral prefrontal, meaning in front of premotor. Cortex. Okay, so that gets a lot of visual input. If you look at, what is that equivalent to in the macaque monkey, it would be something down here, something at the principal sulcus. So like something here. So that's dorsal, lateral, prefrontal cortex in a macaque monkey. So it's in front of a little further in front of the frontal eye fields. But if we go on to the midline, what we see is there's kind of an extra area in there. There's another motor area, and this was originally called M2 for its, secondary motor area. And it turns out this secondary, the secondary motor area has a little frontal eye fields attached to it. It's got its own separate frontal eye field. So this guy is called the dorsomedial frontal eye fields. And this guy is called usually SMA for supplementary motor area. Put those in green to match up with the other one. So this is the dorsomedial frontal eye fields, and this is the supplementary motor area. But as usual, it turned out there's more than one supplementary motor area, so we'll call that supplementary motor area 1, and then there's another supplementary motor area 2. These are all on the midline, so if you look at a midline view, I'll do it over here. So if I flip this guy around, here's the occipital lobe, and there's the parietal, and then here's the frontal, and then here's the corpus callosum like that. And if you look, there's a big sulcus, the cingulate sulcus, and it sort of bends up toward the end. Where are these SMA things? Well, first of all, where is the central sulcus is quite posterior, so this is the central sulcus. And these SMA things are sort of sitting around here. SMA. And the frontal, the equivalent of the dorsomedial frontal eye fields. This guy that's sitting down there too. So let me just label this guy. This is SMA2. So I don't expect you to remember all of these. I just wanted to sort of like lay them all out so we just see what the major players are. Now there's more sort of, eyes, body stuff up here. A little more eyes and body stuff down here. But those are the main chunks. And so what kind of experiments have been done here? So the kinds of experiments that have been done, one experiment which you can see doesn't distinguish kind of muscles from space, is to have a monkey. So there's a long series of experiments done by Georgopoulos, where they had a monkey. So here's a monkey, and its hand is going out and grabbing this little lever, and the lever its little joystick can be moved in different directions. And so they just have the animal move this joystick in all these different directions. And if you do that, you can find that some of the neurons in motor cortex just respond before a particular direction. But the question is it actually coding the direction, or is it coding just a muscle or some combination of muscles? So, which is sort of the same thing as a direction. In order to do that, you have to record from all the muscles and just find out, actually this is just doing the biceps versus it's actually recording this location in space. But there's another set of experiments, if you want to, I have some readings in there, that were done by Michael Graziano. And those are very suggestive that you have something going on where you're actually recording, where you're actually coding extrapersonal space. And so these were stimulation experiments, so long train stimulation. And so if you stimulate the colliculus, you'll make an eye movement. If you stimulate motor cortex, you'll make a hand movement. If you just do a tiny little stimulus, you might see just a little twitch in a muscle. Like if you just go tick, you just see a little twitch. But if you stimulate for a longer period of time, you'll actually get a whole movement out to a particular point in space. And so what what Graziano did was to go into motor cortex and stimulate for kind of a longer train of stimuli to see what happens. And what happens is the animal will make a relatively natural looking movement to some part of extra personal space. So you stimulate there, the animal's just sort of sitting there. You're giving the animal little treats, and it's grabbing little treats. And if you just stimulate there, the animal will just go, wah, and then you turn it off, and then he goes back to whatever he's doing. But interestingly, if you stimulate when the animal is getting ready to reach for something, so what you do is you find a place that causes this movement, okay? And so then you put a little treat, hold a little treat at this position, and then turn on the stimulation right as the animal is getting ready to reach for this location. What will happen is the animal will just go, well, and then go and reach for it very effortlessly. It doesn't seem particularly surprised. It just goes like, I don't know why my hand went there. Now I'm going to grab it. And if you think about what's going on, that's kind of like what happened in some of those superior colliculus experiments. So in those superior colliculus experiments, you have the animal fixating here, and say we put a target up here. So target up here. Now, before the animal has a chance to saccade to the target, we stimulate in the colliculus and say it makes that saccade. Then you turn the stimulus off, and the animal will make the correct saccade. So this is stim-induced. So what's going on here? Well, the animal can do this in the dark. And so what's going on is you did a stimulation. The whole system said, okay, this is where we went to, and so I'm going to update the motor map in the colliculus so that now we can make this correct saccade over to this target. So it has to get information from where the eyes went to and feed it back into the system, and then acquire the target. That's exactly what's going on in this long stimulus train, or do it like, target, stim, before movement starts. It's the same thing. So what happens is you put on the target on, then the animal gets ready to move for it, but you stimulate it and the animal makes this movement instead, and then it makes the movement to the target, which is a different movement to the target than it would have made from the starting location. And so it must have calculated, oh, my hand is over here now, and so I need to update my map so that I can sort of like acquire where the target actually is. Very similar to what, this was done in the superior colliculus. So that's kind of interesting data that suggests maybe there are some kind of motor maps of the kind that we have in the superior colliculus, like different motor vectors, basically, in motor cortex. And one final thing to think about is that these maps are not the same as the somatosensory maps. So, behind the motor cortex, you've got all these somatosensory areas that we talked about, S1. And those areas are, what are those? Those are little patches of skin from all over the place pasted down onto the cortex. You take your three-dimensional body surface and you have to sort of flatten it onto the cortex. And how do you do that? Little segments of fingers and chin and foreheads and stomachs and everything sort of all pasted down there. Motor cortex is different because motor cortex is controlling the muscles, but the muscles are not in the same place. So like if I move my wrist like this, what's doing that? So you can see this muscle over here that's moving my wrist. Now, that's way away from my wrist. It's not lined up with the wrist. What are the muscles that do this? The muscles that do a powerful contraction like this, they're located down here. That's actually where they are. You can feel it. Just like move your fingers like that, you'll feel muscles moving down here. So you often see like, a somatosensory homunculus and a motor homunculus lined up, quote unquote lined up. But it's not quite the same. So, you've got, in motor cortex, you probably have these, like a whole series of maps of different different kinds of motions into extrapersonal space. We don't understand, they haven't been completely mapped, we don't fully understand this, but it's probably something a little bit like the deep colliculus, where you have essentially a map of different movements. And you've got the same deep colliculus problem where we've got to get information about where the movement went to in order to program our next movement, because we don't make all movements from like a starting position. We sometimes grab this and then we grab this. And so then you've got the same kind of problem of updating where your limbs are. Or you can do this in the dark. So if two little sounds appear in the dark, you could make a movement to this guy and then make a movement to this guy. And so you're going to have to do all this remapping business in the motor system as well. Okay, so that was a not very satisfactory overview of tons of research into the motor system. But if you want to look some of this up, at least there's less areas than there are visual areas. That's a positive aspect of the motor system. Not too many. You could almost remember this. Area 4, area 6, frontal eye fields, dorsolateral prefrontal, dorsomedial frontal eye field, supplementary motor, M2. So you could kind of think of these guys together. These guys together are, M2. That's the original M2, or supplementary motor area. Yeah, so there's a little less there. So okay, so any questions about the motor system? A lot of experiments. So there's a lot of experiments in the motor system, especially with reaching and monkeys sort of grabbing at stuff, because monkeys are very good at grabbing at stuff, and just like us, good at grabbing stuff. In rodents, most of the work on the motor system has to do with the vibrissae. So they also, rodents have pretty good hands, and they can also like press little levers with their little rat paws. But there's also a lot of work with the vibrissae, because the vibrissae actually move. They've got muscles that actually move them actively, and so it's actually sort of a motor system. The whiskers of a rat are kind of a sensory system, but they're also a motor system also. That may have something to do with why you decided to paste both of these motor and sensory inputs into the same structure. Well, because what they do, so the vibrissae, so cats, don't have, humans have crap whiskers, cats, they can't move theirs really. But what rats do is, or rodents do, is they got these vibrissae out there, and they're like a high performance touch-based feel in the dark system. And what they do is they rotate them around and sweep them. If you film them and slow them down, you can see that they're sweeping over objects with them, and with every breath. So they're going. So if you film it in slow motion, you'll see that they've got these little detectors that are sticking out of their head, and they're like sweeping over the environment to sort of like figure out what's out there. Where are the objects out there? So they're very high performance little distance somatosensory detectors that can sort of extend out quite far from the rat. If you ever get a rat or have a pet rat and just look at it, you'll see them going. They're just like cycling around. They're breathing like about 7 times a second. So it's quite. They're breathing. Their heart's going. And they're just like doing their somatosensory thing. If you come up to the like if you approach a rat from the top, that's very threatening. But if you approach a rat from the bottom, like, here's the rat, like here, and you approach it, and then they'll say, like, you're kind of a friendly, maybe another rat. And then you can see the whiskers going, and they'll come up, touch you, see what's going on. So, yeah, so they're very active sort of sensory devices, very high performance whiskers. Yeah, I mean, if you've ever seen a rat, you might have noticed the motion, but you weren't paying attention to it. But if you look close at them, you can see that those whiskers are just going crazy all the time, because they can figure out what's going on pretty well in complete darkness, and they extend out quite a long distance. They've done experiments on rats also with remapping, like what happens if you glue two whiskers together, doing bad things to rats. So if you do that, they have a representation of each whisker separately. There's a little array of whiskers. And so if you glue two together, the representations will fuse because now they're picking up the same thing. Yeah, so if they completely lose whiskers, if one just drops out, another one will replace it. But if they get in a rat fight and like somebody bites their whisker and damages it, then they'll have to remap the ones that are left. What? Oh yeah, it will always remap. Yeah, no, they'll always. It's an active spatial detection mechanism, and it's not just whiskers here. They have sort of, whiskers over their eyebrows also, and they've got whiskers. There's some whiskers coming out of their wrist, and it's like a whole body system. They don't have any whiskers on the little rat tail though. Okay, so now let's quickly go over the class. So we start off with visual. So we'll start with visual. And so what do we do in visual? So we did a lot in V1, and so we looked at all these different areas in V1 that were like the blobs, and there's like several layers here, and there's orientation columns. So we kind of like went over some of the basic layout of the cortex, and what are the input layers. Remember, there's the input always comes to layer 4, almost always comes to layer 4, and then from layer 4 it sort of like goes above and below layer 4, and then layer 5 is a big output layer, and layer 2, 3 goes to the next area. So that was the sort of general V1. And then we talked about in V1, we also talked about simple and complex. So that was another. And we said that, complex are not that complex. It was like simple, slightly less simple, and then hypercomplex were just even a little less slightly simple, but still pretty simple. And so that guy just doesn't work. So what was the simple and complex business? So the simple and complex business mainly was, how do you make a simple cell? You take a bunch of retinal or lateral geniculate on cells and line them up. And here's their surround. They might have an inhibitory surround. And then you take a bunch of off cells. I'll put those in red. Make a little row of off cells. And, maybe there's another row of on cells. You could, sometimes you have 2 rows, 3 rows. But basically, if these guys are off, meaning they like it when it gets darker, and then these guys are on, they like it when it gets brighter, then you have a simple cell. So here's the simple cell. So that thing is the simple cell. And it's a way of detecting orientation because you've sort of elongated these little rows of on and off cells. And it's good at detecting orientation, but one of the problems it has is it's not very good at detecting an edge, a general edge. And the reason is because if it happens to be a light edge, and the light edge happens to be sitting over the off region, well, it doesn't care about that. You won't get any response because off only likes when it gets darker. And so it sort of gets confused by the bright versus dark. And so if I were to move an edge, if I were to move a light edge across here, when it went into the off part, there would be no response. If I went across the off part, it says, I don't care about that. It got lighter, but I like when it gets darker. And then when you hit the on part, you'd get a response. And then when you hit the off part, you'd get no response again. But what if I had a dark edge? I don't have any dark edge handy, but if I had a black edge against a white background and moved it in here, then I would get a response when I hit the off cells because it got darker, and they like it when it gets darker. And so you can see it's kind of confused. So the complex cell is pretty much the same thing, but somehow we've set it up so that there's off and on everywhere. So if you turn on a little light at a spot, you'll get a response if you turn it on or you turn it off. And so somehow by combining separate simple cells, we can end up with this kind of a cell that just detects any old edge. And so this cell is better for detecting an edge, but it's lost some information. It doesn't know whether it's a bright or a dark edge. It doesn't know exactly where the edge is. So that was the basic distinction between a simple and a complex cell. So the complex cells combine more information across space, but then they kind of lose, is it a dark or a light edge? But maybe you just want to know that it's an edge, and you don't care whether it's dark or light. So that's the complex cell. And then the hypercomplex cell was just slightly more complex, the slightly more complex cell. And that's the one that kind of looks at things that it could be a simple or a complex cell, but it's a cell that basically has some inhibition at the end. So that's hypercomplex. There's some inhibition. So that means if the object gets too long, it turns off the cell, and so the cell will like sort of a line coming to an end. So that's a hypercomplex cell. So it might be selective for something that looks like that. So like a long line that comes to an end. And there's a lot of stuff like that. Grass blade sort of looks like that. There's a lot of different lines that come to an end out in the world. So that was simple and complex. Any questions about simple and complex? And we talked about the dreaded aperture problem. So the aperture so what is the aperture problem? So the aperture problem is we've got these detectors that we call Reichardt detectors, and what they do is they essentially look through the world at a tiny little bit like that. And here's I'll draw the Reichardt detector down here, and it's got one spatially offset and also delayed detector that might sort of go to this part here, and then a not delayed detector. Let me pull that one in a little bit because it's all within that receptive field. So this thing goes to this part of the field, and this thing goes to this part of the field. So we've got that little detector in there, and it's got a spatial offset. It adds them together. It's got a spatial offset here. So those detectors are separated in space, and one of them is delayed. That's what a Reichardt detector. Reichardt detector. So if you make something like that, it's going to have an aperture problem. And what is the aperture problem? The aperture problem is it can only pick up motions perpendicular to the contour. it can't pick up because of the way it's set up. It's got two offset detectors. So if something moves parallel to the contour, it doesn't know. And so what does that imply? That implies that any of these motions could have happened. So, maybe it went that way, maybe it went that way, who knows? We don't know. Any of those motions are good. And so that's the aperture problem. And it comes from the fact you made a detector to detect motion in one direction. Yeah, question?
Oh no, you were literally just about to answer it. I was just gonna sum it up to make sure I'm understanding, but I think it just clicked. Okay, so because of the way you made the record detector, it can only detect one motion.
Yeah, one direction, yeah.
Yeah, one direction, but it doesn't take into account all the other directions the object or whatever it is could have made.
Yeah.
Yeah.
Yeah. That's the aperture problem. It can't detect any sliding motion this way. But if there's some sliding motion that way, it could be that this whole thing is moving up instead of moving this way.
So if we were to say you just draw record detectors in the motion, you know what I mean? So if we were to draw the detector a certain say it was opposite or different way, would that be wrong, or is there only one right way?
So basically this detector just detects that local motion. So the little thin arrow, this was the local motion, meaning just the motion that the Reichardt detector can detect perpendicular. That's what we mean by local motion. The pattern motion is the actual object motion, which might include some sliding along that contour. And unfortunately, any of these pattern motions could have caused that local motion. So that's the bottom line. We detect one local motion, but all these different thick arrows could have caused it. So this is the actual pattern motion, the thick arrow.
So that's the direction it's actually going.
Yeah, that's the direction the whole thing is going. So maybe there was an edge here, but the edge was moving vertically. So like my hand, say it was just detecting this piece of my hand. Well, my hand was going up.
Oh, yeah.
My hand is going up, but this detector is going to say, I can't see that because I can only pick up a motion that's perpendicular to the contour because that's the way my detectors are set up. Yeah, so it's, like I said, this was discovered, it's a little non-obvious because these, the mathematical guys at MIT set up some video and they said, whoa, we can't tell which way we set up these little detectors, can't tell which way it's going. So how do we solve that? So, we solve it by, we have to sum across space. So if we have an object that's moving in that's the actual direction it's moving. What's going to happen is, if it actually moves in that direction, then we would detect that motion at that contour, and we would detect this motion at that contour. And so those are slower motions, and they're off to the side. But if we can somehow kind of sum across those two, then we can find out what the real motion is. And how do we do that? We said it was a voting scheme. So you have for that actual motion, there's going to be a whole series of possible local motions consistent with it. This is kind of the converse of this picture. These two pictures are sort of like the inverse of each other. So this one is we have one local motion. What are the possible pattern motions? This is, we have one pattern motion. What are the possible local motions that could have caused it? And so we would have a whole set of these guys, and there's going to be some things in common between them. So it's not going to work if we just have one, so here's all these possible local motions for this pattern motion. And if we happen to get this guy over here as our it's going to activate two of these guys, right? Both of these are the same. These are the same. That's just the same way of looking at this. These two guys, this guy and this guy are both possible pattern motions that could have been caused by that locomotion. So if you just have one detector, it's not going to work because it's going to activate multiple ones of these. And so what we have to do is have a whole bunch of different angles, and then what happens is we will find out which one gets the most vote. But multiple ones are going to get votes. It's just the one that has the largest number of votes. Yeah.
Is the largest number of those across all detectors?
Yeah. Yeah. So it has to sum across space. So this is across space. So it has to slightly damage the map because it needs to integrate information across space. So there was that, and then we went to MST, and remember MST was if you go to MT and you rotate something around versus rotate it around the other way, you can't tell the difference. And so what we have to do to make MSTD is we have to sum across even more space to try to figure out, is the whole world rotating or is it expanding because something is coming toward you, or is it contracting because you're, say, backing up?
So this is a contraction problem or something like that?
Yeah, so if I'm looking at the board and I back up like this away from the board, it just looks like I'm backing up. But if I went to every little piece of the stuff on the board and measured what direction it was moving on my retina, it would all be moving inward. Because when I'm close like this, it's spread out across a lot of my retina. When I'm far away, that thing is much smaller. And so everything just went in when I did that. And vice versa, if I just approach something, everything expands. MT can't see that kind of stuff. So MT cannot detect whether it's expanding or contracting. How do you do that? Well, just take an MT receptive field like this and, try some expansion and then try some contraction. No difference. It might respond a little bit because, if MT happens, if this MT neuron happens to like up, well, there's a little bit of up in here in this rotating pattern, and there's a little up in this rotating pattern, and there's a little up in this expanding pattern, so it'll respond a little bit to all of them. So MT would say, yeah, all of those are good. I can't tell the difference because they all got some up. So here's some up, here's some up, here's some up, here's some up. Can't tell the difference. So that was MST, and it has to do it by sort of summing information across space. So then we went to Somato. And there's not too much in somatosensory. The main thing we did in somatosensory was all the different receptor types. So we had lots of different receptors. Yeah, question?
So going back to MSTD, you're saying that summing across space for MT is what creates MSTD, right?
Yeah, so what you would do is you would take, so MT just assumes, you can sort of say, MT assumes the whole world just moves as, the whole world is a pattern that is moving in one direction. That's kind of what MT's assumption is. And so you take a bunch of MT neurons and look at different parts of this rotation pattern and then assemble them into some kind of template of rotation, and then that's capable of actually picking up the rotation. But the individual MT inputs can't. They can only pick it up by sort of looking at different parts of the rotation with MT inputs. Okay, so somatosensory, we had all these different receptor types. there was, touch receptors, Pacinian corpuscles, that's a touch receptor, Ruffini endings, Merkel discs, the Meissner corpuscles, hair cells, all those guys were touch. So we had, touch, and then we had pain and temperature and, pleasure, like little stroking guys. And then we had the muscle receptors. And the muscle receptor was a little more complicated. We had the muscle spindles that detect length, and then the Golgi tendon organs. And so these guys were detecting length and force. These guys, that's Golgi. And this is muscle spindles. Yeah, so these guys, and remember how the arm worked. Like these muscle spindles are bolted down to the muscle. So here's the muscle like this. You got this big muscle, and the muscle spindle is actually sort of bolted down to the muscle like that. And it's got a detector. The 1A ones have a detector in the middle, and then they got their own little muscle. And then it's attached to the muscle. So how does it work? Well, if the muscle contracts and nothing happens, this detector is just going to go slack, and the detector can't detect slack. And so the way it works is you send out a signal, contract the main muscle, and then at the same time contract these little spindle muscles. And if everything goes right, the muscle spindle detector doesn't get stretched. But if the object is heavier than you're expecting, this muscle won't contract as much. And then what will happen, this guy is the muscle spindle muscle is the prediction of like how short is the muscle supposed to get. And it didn't get short enough. And so then this guy will get stretched a little bit. During contraction, yeah. And so it's weird, like what causes the signal? Well, the muscle spindle muscles cause the signal. But really what they're doing is they're making when you contract these guys, these are the gamma ones, when you contract those guys, that's the prediction of what how short the main muscle is supposed to get. It's a prediction of how short this guy is supposed to get.
So gamma is the one that predicts how short the muscle is supposed to get, or how short the main muscle?
Yeah, how short the main muscle is supposed to get. So it's predicting how short the main muscle is supposed to get. And then the alpha motor neurons, those are the ones that are actually controlling those are controlling the main muscle. So these guys are just synapsing just on those little muscle spindle muscles. So basically, so this is the main here. So basically, those guys will say, I'm expecting the main muscle to get such and such shorter. Yeah?
So, but the muscle spindle, so that's like the muscle in the muscle, and that's also predicting how short the muscle is supposed to get, when the gamma is the main muscle.
No, the other way around. No, the gamma is the muscle spindle muscle. Gamma is the muscle spindle muscle, and the alpha is the one that goes to the main.
Oh, gotcha. Thank you.
Yeah, so this is, let's just label this guy here. So this guy is muscle, spindle, muscle. Yeah. That's the contract, yeah.
And the gamma, the Golgi?
Yeah. Not Golgi. Golgi's down here.
Oh, okay.
This is Golgi, yeah, there.
So the muscle spindle, which is the green part, that one's like, we were supposed to contract this much, and now it's being stretched.
It gets stretched a little bit, yeah. So if it doesn't contract enough, it gets stretched. And that's signal, right. So that's a signal that comes back into the nervous system that, that's the one that says you didn't contract enough.
Because of force.
Yeah, because the thing was heavier than you expected. Yeah, so I mean, again, this is something you don't realize you're doing it, but every single time you pick up something, you have estimated how heavy it is. It's just like, that's light. I don't need much force. Whereas like the table, that's heavy. I got to do a lot more. So you're constantly doing that. Okay, so that was somatosensory. Okay, so then we had auditory. And so there were two main things we did in auditory. We did basically owls and bats. And so the first thing was, the very first thing we did were auditory maps are 1D. So remember, versus 2D, because all these other maps like visual, somatosensory, they were 2D maps. Like you have two different directions on the skin, you got two different directions in the visual field. But the auditory maps are 1D because it's just, so that's the first difference. They're different because of that. And then we went, into the owl, the owl brainstem. And so this was a problem where we have the problem of we've sorted things into frequencies. And so the problem is, should we match this guy, with that one? Is that the delay? Or is that the delay? Or is that the delay? And how does this work? Well, we have some neurons that fire at the peak of the sound wave, and they come into this nucleus and from the left and the right side, and then you've got this set of coincidence detectors, and then they just match spikes. So here's that nucleus laminaris structure like this, NL, and frequency is going that way. And so perpendicular to frequency, you just match up spikes. So here's a spike coming in, there's another spike coming in, and say there's only one coming in from this side. They're just coming by like that. You just match them all. And so this nucleus does not solve this problem. It just matches this one with this one and this one with this one. And so it will fire different cells. Multiple cells are going to get fired. It's ambiguous. And then the way we solved it is we said we've got to look across frequency. So if we go up to the inferior colliculus central lateral part, it kind of looks pretty much like this thing. So this thing along the horizontal axis, it's got this delay business. So that's the delay business. It's still there. So this is the characteristic delay. And then along this axis got frequency. And so what we said is if you only have one pitch, you might get several locations in the nucleus that will fire because you matched this guy with this one, and you also matched it with that one. And so you'll end up with two delays. And so you're hosed. If you just got one pitch, you can't figure it out. So you need at least one more pitch. And so if you get one more pitch, you might have something like this in the nucleus. And then if we sum across all the columns, we can find that this column gets the most votes. This guy doesn't get as many votes. This guy doesn't get as many votes. Yeah, it's democracy, right? And so this turns out to be the real delay. So this is the real delay. And if we have like a whole bunch of different frequencies, which is the best of all possible worlds, what would this pattern look like? Well, it would sort of fill the whole thing in, and there'd be these kind of stripes, stripes going across the nucleus like that. That's if we have like all different frequencies. So that was the trick, and it could be, the owl is trying to just figure out where the mouse is, but we could also use that as humans to try to focus on sounds that are coming from a particular place. like to do, auditory, spatial attention. So that was the owls. And then last but not least, in the last negative 15 minutes, we will talk about bats. And so what was bats? So bats are just trying to eat bugs, but it turns out they have this problem of figuring out the Doppler shift. And so the bat problem is we need to basically figure out, is the bug flying away from me, or is the bug am I approaching the bug? And so what the bat needs to do, and it's constantly changing its pitch depending on how fast the bug is flying in order to get back the signal at the sensitive frequency. And because it's constantly, I've just drawn one pitch here, but it's constantly changing its pitch, and so it needs to calculate the Doppler shift. And what it does to do that, it compares the frequency of one of its outgoing to the frequency of one of the echoes. And so it's using this to calculate Doppler. So that's what the bat's doing. But what is the problem for the bat? The problem is this frequency might be different. The frequency, it could be, if he's flying toward the bat, maybe it's 30 kilohertz. But if the insect is running away, it might be like 28 kilohertz. That's too low. And so it's constantly fiddling with its voice to, like the next call out might be a different frequency. And so what is the bat problem? The bat problem is, I want to know whether I've got this spacing between the echoes, but I don't care about the absolute pitch. So I also want to know if I've got that spacing. That's one Doppler, and I'll exaggerate the difference, but there might be another Doppler that looks like this. The difference wouldn't be that big. And so it doesn't care like what the actual frequency is. It just wants to know, is there a big difference between these or a small difference between these, and throwing away whatever the frequency you're at. And we said that that's a similar problem with speech, because if somebody has a very small vocal tract, then when you look at their formant bands that indicate different vowels, say like this indicates E, but if you take another person, a person 2 with a small vocal tract, like a kid or a woman compared to a large man, their E is going to be up in frequency, like that. And so what we want to know is how far apart are the two frequency bands, but I don't care what the actual, so these are both E's, but if we made an ah, so this is person with big, vocal tract person with small. So if we made an ah, it might look something a little closer together like that. So there's a big person ah, and here's a small person ah. And so what we want to do is get those two apart from each other. So here we say these two frequency bands are close to those formant bands. So this is F1, F2. And then here they're farther apart. And so it's the same problem. It's the same problem that the bat has. The bat's trying to just figure out the spacing between these things, but doesn't care like the offset. Now, there's a separate issue. Where do the bands come from? In the first place, it's different than the bat. So, we said that if this is the pitch of your voice, and I'm saying E, and I'm going E, sounds like E the whole time. And why is that? Well, all these harmonics are going up because my voice pitch is going up. But what the formant is you set your tongue in a particular position. And what that does is that just lets through whatever harmonic happens to be right at that frequency. And so you get a band like F1. And then if you set the lower part of your tongue that's in your throat differently, you'll emphasize, a different band. And so that's different than the bat, because the bat is just using the harmonics directly. Here, we're kind of filtering the harmonics. We're taking all the harmonics of our vocal tract. This is the, F0, which equals first harmonic, unfortunately. So this guy is the first harmonic. So unfortunately, F0 equals the first harmonic. And then the higher bands, frequency bands, aren't harmonics. They're just like the tuning of your mouth. So that's why E, it's just because my mouth is allowing those single frequency bands out. But the problem is I got a big vocal tract. You take somebody with a smaller vocal tract, the whole thing gets shifted up, and you've got to instantly be able to recognize it. Yeah. Yeah, it's a completely different function. Yeah. Yeah, all we were saying is, what is the actual kind of signal processing problem the auditory system has to solve? And so they figured out, like in bats, you've got this little, you have this map of frequencies, like, 30 to 30, 32, and then like 90 to 96. And so really what all the bat does is it says, are the two frequencies along the diagonal? That would be no Doppler shift. Or are they sort of above the diagonal or below the diagonal? And we've got we probably are doing the same thing in our auditory cortex, where we're saying, like, if you plot, formant 2 against formant 1, you'll get different vowels will kind of look like this, depending on, so if you've got a really small person, they'll have a relatively high frequency. If you got a bigger person with a bigger vocal tract, they'll have a low frequency. And somehow we've got to distinguish those from each other. It's similar to the bat. It just needs to distinguish those from each other. So it's the same computational problem, completely different function. Yes.
So for the Doppler shift, you said, like, if it's perpendicular, it's not if it's above or below, that's the Doppler shift. If it's above or below perpendicular.
Well, this would be like a zero Doppler. So if you're along the, so that would be like the bat and the bug are moving the exact same speed. So there's no Doppler shift. So if the bug's accelerating away from the bat, then it's going to get lower, because the way, as it bounces off there, it's going to come back a little slower. If the bat's closing in on the bug, which is normal, then the echo is going to be higher. Yeah, so if the outgoing went out at, say, 30, and instead of 90, you got back, say, 93, like you got something over here, that means, positive Doppler, right? Because the bat's closing in on it, plus Doppler. And if you sent out 32 and you got back 90 instead of 96, that's going to be, negative Doppler down here. This way is negative, that way is positive. Okay, so I think we covered everything. Any final? I haven't made the test yet, but any final questions? off the dome. Well, I mean, I won't ask this question, but I've occasionally asked the question. Say you have two little apertures, and say you got this. So say there's an edge here, and there's the same edge here, and you got two signals. Like this edge gave you that signal, and this edge gave you that signal. Okay. Is that one or two objects? It's an aperture problem, but the question is, could these be part of one object? And the answer is no, actually.
Are we talking about like the direction, like when you say could they be part of the same object, you mean like the object is the movement of the
Yeah,
object?
yeah. Could it be the same? Yeah. Could it be could it be two parts of the same object? And the answer is no. It can't be because of a rigid object. So it can't be part of a rigid object because if you have the same Reichardt detector, it's going to pick up the same locomotion. You can't have like a faster thing coming out. Similarly, like if you had something like this, like if you had one edge. Yeah, like this couldn't be a single object either. You can't have like two, opposite directions. That's not allowed. Is that maybe there could be one similar to that one, I think. Well, here's one that I sometimes put on an exam. So say you have two edges like this, and it's one object. I told you it's one object. And you got like a little signal coming off of that one, and you got a signal coming off of that one. What direction do you think it's going? Yeah. Yeah, I mean it's going to be going up, but how would you calculate it? So what you would do is you would say it could be that one, it could be that one. Yeah, it could be that one, and then what's going to go on with this one? Could be that one, could be that one, could be that one. And so what you got to do is you got to find one that's the same arrow. Yeah, so it's like this one right here is the same arrow as that guy. And so, it's actually going, slightly that way. Easy peasy, yeah.